Hi dear viewers, I am Kalaivani from SNS College of Pharmacy and Health Sciences working as an assistant professor. So today we are going to see about a isolation process of some chemicals. So first we'll go with the Diosgenin from Dioscorea. So nature has always been the greatest chemist hidden within medicinal plants or molecules that have transformed modern medicine, and one such powerful molecule is Diosgenin, a naturally occurring steroidal sapogenin obtained from the tubers of Dioscorea. So in this lecture, we will understand from the very beginning to final step how Diosgenin is isolated from Dioscorea, including its principal, procedure, and significance. So, isolation and detection of Diosgenin from Dioscorea. Diosgenin is a steroidal saponin obtained by initial hydrolysis of dioscin, which is present in the tubers of various Dioscorea species such as D. deltoidea and D. floribunda D. composite. So it belongs to the family Dioscoreaceae, and it is present to the extent of 2 to 5% in Dioscorea tubers, and Diosgenin is an important starting material for the synthesis of some of the steroidal hormones, and it is converted to 16 dehydro preginolone acetate, which is used as a substrate for various types of steroidal drugs such as corticosteroids, sex hormones, oral contraceptives, spironolactones, etc. Diosgenin is used as a pharmaceutical aid for the synthesis of various steroidal drugs. And coming to the next, that is the principle of the Dioscorea. Uh, Dioscorea belongs the properties. It is white crystalline and solubility, organic solvents and acetic acid, and chemical constituents dioscin and diosgenin. So, it is a genus belonging to the family that involved we have seen, and the principle behind the isolation of diosgenin is acid hydrolysis of saponins. In Dioscorea, diosgenin exists in bond form as a glycoside called dioscin. When treated with dilute mineral acid, the sugar moiety is removed, releasing the aglycone diosgenin, which is insoluble in water and can be separated and purified. So we required some dried tubers of Dioscorea, and the chemicals we require dilute HCl, that is hydrochloric acid, or sulfuric acid, ethanol, distilled water, organic solvent, that is like chloroform or petroleum ether, whatever we can use. Then apparatus, round bottom flask, reflex condenser, heating mantle, filtration setup, separating funnel. So the first step is preparation of the plant material. So we took the Dioscorea tubers, cleaned thoroughly, dried it in the shade, pulverized into a coarse powder. This increases the surface area of efficient extraction. And coming to the step 2, it is acid hydrolysis. The powdered tuber is transferred into a round bottom flask, and to this we will be adding dilute hydrochloric acid or sulfuric acid, and the mixture is then heated under reflux for about 4 to 6 hours. And during this process, steroidal saponins undergo hydrolysis, sugar pods dissolved in water, and free diosgenin is liberated. So now coming to the third part, that is step 3, cooling and filtration. After refluxing, the mixture is cooled to room temperature. The solid residue containing diosgenin is filtered. The filtrate contains sugars and impurities which are discarded. And washing the residue is washed repeatedly with the distilled water until it becomes acid-free. This step ensures removal of residual acid and soluble impurities. And coming to the drying part, and the drying and extraction. This is the step 5. The wash residue is dried completely, extracted with an organic solvent such as chloroform or petroleum ether. Diosgenin dissolves in the organic solvent due to its non-polar steroidal structure. So next, the concentration of the extract. I think this is the last step, and this organic solvent extract is filtered, concentrated by evaporation, and on cooling, crude diosgenin crystallizes out. So purification of diosgenin was done after this extraction process, and crude diosgenin is purified by recrystallization using ethanol or suitable solvent, and the pure diosgenin appears as a white to pale yellow crystalline powder. So we need to identify whether it is diosgenin or not. So we will be taking for the identification test. The first one is Lieberman-Burchett test, and here we will be observing some positive green coloration which confirms the steroidal present in the nucleus. That is in the diosgenin particles. Melting point. Pure diosgenin melts at approximately 204 to 207 degrees Celsius. See how much high it is. And the yield and storage, it depends upon the plant source and efficiency. And isolation of diosgenin demonstrates glycosic hydrolysis, highlights industrial drug synthesis, and the classical pharmacognosy practical. So thank you. Understanding this isolation not only strengthens conceptual clarity but also builds a bridge between natural products and life-saving medicines. Thank you so much. Hi dear viewers, I'm Kalaivani from SNS